Namaste students, हमेशा खुश रहो बिटिया. I am JV's Dr. Neeraj Dholia, Faculty of Agriculture and Veterinary Science, Jayoti Vidhyapeeth, University, Jaipur. So today we are going to discuss a practical session, that is estimation of protein by Lowry's method, and it is an important practical, and the subject which it is related with is metabolism and bioenergetics lab. So let's start. So the aim of the experiment is estimation of protein by Lowry's method. Let's talk about the principle. So the peptide bond in the polypeptide chain reacts with copper sulfate in an alkaline medium to give a blue colored complex. So in addition, tyrosine tryptophan residues of proteins cause reduction of the phosphomolybdate and phosphotungstate components of the following calculative reagent to give bluish product, which contributes towards enhancing the sensitivity of this method. So the materials which required here is a reagent A, which is a 2% sodium carbonate in 0.1 normal sodium hydroxide. Then reagent B, which is 0.5% copper sulfate in 1% potassium sodium tartrate, prepared freshly by mixing stock solutions. Alkaline copper solution, mix 50 ml of reagent A and 1 ml of reagent B prior to use. So diluted following reagents, that is dilute following calculated reagent with an equal volume of 0.1 normal mMH. Then standard dissolve 50 mg BSA in 50 ml of distilled water in a volumetric flask. Take 10 ml of this stock solution and dilute to 50 ml in another flask. So for working standard solution, 1 ml of this solution contains 200 microgram of protein. So apparatus and glassware which is required is test tubes, pipettes, and the colorimeter. So procedure, how to proceed this experiment. Pipette out 0.2, 0.4, 0.6, and 0.8 and 1 ml of working standard in the series of labeled test tubes. Then pipette out 1 ml of sample in another test tube. Then make up the volume of up to the 1 ml in all the test tubes. A tube with 1 ml of distilled water serves as a blank. So now add 5 ml of reagent C to all the test tubes, including the test tubes labeled blank and unknown. So mix the content of the tubes by vortexing or shaking the tubes and allow to stand for 10 minutes. Then add 0.5 ml of reagent D rapidly with immediate mixing well and incubate at room temperature in the dark for 30 minutes. Now record the absorbance at 660 nanometer against blank. So then plot the standard curve by taking concentration of protein along x-axis, absorbance at 660 nanometer along y-axis. Then from this standard curve, calculate the concentration of protein in the given sample. So result will be the protein in microgram protein per ml of sample. So this is the table and graph you should make, that this is a standard curve calculation, and this is a standard curve. In the x-axis is the concentration of protein, and in the y-axis the absorbance at 660 nanometer. So thank you. Hope you are satisfied with this tutorial session. If you have any query, please write in the comment box. I will resolve your query. And this session is powered by Digital Version 2, Jyoti Vidhyapeeth, Women's University, Jaipur. Thank you.